Right, so we will be continuing the conversation of macromolecules by looking at proteins. And again, looking at proteins, we see that there are a lot of functions as it relates to proteins. So the next couple of slides will be some pictures and then we'll go into some context. So proteins, they're more than 50% of the dry mass to most cells. We see that they are in a lot of cells. They have storage, transport, they allow cells to talk to each other, they have movements, and we see that they also help defend us against foreign substances. I will have you note, I'm not going to read through each of these, except note that there are enzymatic proteins, defensive proteins, storage proteins, transport proteins, and we'll see that there are 8 of these. These are the 4 of the 8. These are the last 4. There are hormonal proteins, receptor proteins, contractile motor proteins, and structural proteins here. And then enzymes. Enzymes is a type of protein, and I will say this often where enzymes are proteins, not proteins are not enzymes. And we'll see that enzymes are a type of protein that acts as a catalyst to speed up. And so they can help perform the functions repeatedly. So then there are membrane proteins here, and you can see that there are a lot of membranes, and this is looking at the plasma membrane. So there are transport, receptors, intercellular, cell-to-cell recognition, and enzymatic. We see here what a transporter is, a receptor. It can bind to a protein and send a message. There are enzymatic that will help turn in one molecule into another, and then there are anchoring proteins. Now with proteins, it's really like we have to consider amino acids, and there are 20 amino acids, and these are the monomers of proteins. The amino acid structure is the central carbon atom. There's an amino group, a carboxyl group, a single hydrogen, and a variable R group. And so we see here with this picture that we have an alpha carbon and an amino carboxyl and a side chain R. Now, one might wonder, what are all the chains? Well, here are all of the examples: glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, proline, serine, threonine, cytosine, tyrosine, asparagine, glutamine, aspartic acid, glutamic acid, lysine, arginine, histidine. And you can see that they have all these different characteristics. Anytime they have different functional groups, they have a different characteristic that they will show. Excuse me. So what we'll see here is we have a peptide bond. And when we have a peptide bond between these amino acids, we see that we have side chains and we have these new peptides, this dehydration synthesis that will have this amino acid peptide bond and a carboxyl end, a C terminus. So we'll have an N terminus and a C terminus on an amino acid. So amino acids are joined by dehydration synthesis using a peptide bond. Proteins are can form polymers of one or more long unbranched chains, and each chain is a polypeptide. They are biologically functional, so they have function. Shape determines function. They are linked by peptide bonds. So each functional protein has a unique shape, and again, shape determines function. So here we see a ribbon molecule versus a space-filling molecule or model, and you'll see that the groove of where the shape is, and we'll talk about the shape of proteins. The ribbon versus the space is going to be two different, it's the same, but it's shown differently on different diagrams. And here we see this is an antibody protein and a protein from a flu virus, and how they actually can help bind together. So these polypeptide chains are going to fold in various ways to form a shape. And so between these shapes, they can form different bonds. So they can have a hydrogen disulfide ionic or even a van der Waals attraction, which you remember from our chemistry chart, and even a hydrophobic exclusion. So we can have the shape of a protein will help determine its function. There are 4 levels of the protein structure. So here we have a primary structure, And these are like beads on a string. Remember, we have this amino acid, we have an amino end and a carboxyl end, and we see here that they are numbered. That's the number of amino acids to help us understand the complexity of it. And each of those letters is representing an R group. And so this is the chain of quote unquote pearls. What we'll see here is the secondary structure. The secondary structure is going to interact with the peptide backbone, which is the hydrogen bonds. So we'll see here we'll have an alpha helix and a beta pleated sheet. The alpha helix is a coil, the beta pleated sheet is folded. And you can see here how we have this secondary structure, beta pleated sheet, and an alpha helix. So we have the beta strand is shown as a flat arrow pointing toward the carboxyl end, and then we have hydrogen bonds between the chain of pearls that will allow it to be an alpha helix or a beta plated sheet. This is the secondary structure. Then we have the tertiary structure, which is a final folded shape of a globular protein. And this is where we will see these without R groups, and these disulfide bridges will reinforce this protein structure. And so here we have this polypeptide background, but now we're going to see how these are going to interact among each other. So you can see how we have a disulfide bridge here, we have an ionic bond and hydrogen bond. The disulfide bridge is not something that we saw in the other two levels, the primary or the secondary level, so we see it here in the tertiary level. So this is a tertiary structure combining the primary and secondary structures to make this tertiary structure interaction. And then finally we have this quaternary structure. This quaternary structure is an arrangement of individual chains or subunits in a protein with two or more polypeptide chains. So to kind of compare here, we have a primary structure that goes into a secondary structure that goes into a tertiary structure, and several tertiary structures going into a quaternary structure. Here again showing what these look like. Now let's talk about the application of this. Looking at this secondary structure, we have fibrin, keratins, collagen, and elastin. And collagen is going to be important for our tendons and skins, and it's going to be very similar to 3 polypeptides coiled like a rope. Hemoglobin is going to be 4 polypeptides, 2 alpha and 2 beta chains. And here, one of a spiderweb arrangement. So researcher exposed the spiders to different classes of drugs, and it showed how the spiders would spin different examples of silk, or spin different examples of their web based on the drug that they were exposed to. So it's really interesting to see how this drugs specifically can interact with the protein expression of spider webs, and we know that drugs impact our human body. All right, so these are some applications of where we would find fibrous proteins versus globular proteins. Fibrous meaning that it's insoluble, globular meaning it's soluble, so we can see that these are found in our bodies and in our blood in different ways. And then we have collagen. Here's what collagen looks like. And then we have hemoglobin, and then these are red blood cells. And then when we look at the application of proteins even, we can see that we have something like sickle cell disease, and this is a change in the primary structure. So this is the structure and the ability to function. So sickle cell disease is an inherited blood disorder in a single amino acid or the substitution in the protein hemoglobin. And so what we see here is I want you to see on the chart on the left-hand side, we have normal hemoglobin, and going down, we can see these amino acids in the primary structure. Here's a sickle cell hemoglobin, and this is why it's a genetic characteristic that we need to test for, because when we go through here, we look at the primary structure, we can see at number 6 that the GLU was substituted for VAL. And what that's going to do is that's actually going to expose a different region of the beta subunit of hemoglobin that will allow this molecule to shape differently into this half moon shape or the sickle cell shape. And so it's going to be really important to be tested for that. It's a requirement in the NCAA to actually be tested for this to be able to participate. Oftentimes now, babies that are born in hospitals are tested for this. Then we have motifs, and this is where we have different elements of secondary structure seen and used for a variety of and many polypeptides, and it's useful in determining the function of unknown proteins and to be able to study that. And then we have domains and multiple domains that perform different parts of this protein's function. And so then we also have chaperones, and chaperones will help proteins fold correctly. And this is where, when we don't have this correct, that we end up getting a lot more, this can indicate different diseases. So cystic fibrosis is a hereditary disorder, and we know that proteins appear to have correct amino acid but doesn't fold correctly. Another aspect, and this is usually a test question, talking about the native structure versus being biologically inactive. So what can also allow the proteins to lose its structure is to actually have it go through these environmental conditions that will quote unquote denature it. And denature it will actually, when it changes shape, it loses function. And so things like pH, temperature, or different solutions can denature proteins. This was one of the research aspects that I was studying when I was an undergraduate student, is we would actually denature proteins, then we would try to renature them, and we would want to see what part of it would actually renature the protein, or what if the protein would be able to change out of the denatured state. So we would actually run tests looking at denatured and renatured, the native structure of the protein, denatured protein, and the renatured protein, to see if there was any differences in the results as we exposed our protein to different conditions. Again, protein, looking at Jello protein, we see that Jello is an example of that. Here's another protein. The reoccurrence of renaturation of a denatured protein is not common. This is also really important when we start talking about little kids and fevers. Temperature being a protein, we really want to be aware of any fevers that are over 104 degrees, because after or above that, we can start to see some type of brain damage at 105 and higher. So we really watch this with little kids and even adults, because it impacts the proteins, and it impacts the proteins in their body and their brain. So then when we talk about nucleic acids is a final topic where we see that this amino acid that we were just talking about from the polypeptide is going to help us give us this unit of inheritance called a gene. And genes are actually made of DNA, which is a nucleic acid made of monomers called nucleotides. There are two types of nucleic acids. There is deoxyribonucleic acids and ribonucleic acids, and we're going to talk about both of them. So a nucleic acid is a polymer. The monomer is a nucleotide, which is made up of pentose sugar, phosphate group, and a nitrogenous base. You do need to know that. You need to know all of it, but those are some key characteristics. And the key characteristic between DNA and RNA is this pentose sugar. In DNA, it's deoxyribose, and in RNA, it's ribose. So we can see here quickly how we have a polynucleotide, or a nucleic acid. And this chart gives us a lot of information looking at the different nitrogenous bases here on the right, and the purines versus pyrimidines. We have 3 pyrimidines, 2 purines, CTU, so cytosine, thymine, uracil. Uracil only occurs in RNA, thymine only occurs in DNA, and then we have adenine and guanine, which are purines, and we say that those have 2 fused bases. And we'll talk about what that looks like shortly. A couple of things else about this sugar phosphate backbone is we have a phosphate on the 5' end, and we have over here this OH on the 3' end. And the primes mean the carbons, but we will use it also to help us understand the orientation of our strand so we can understand what bonding will look like. So here's that picture a little bit maybe clearer for you to see. A nucleoside, we can see that terminology, and we'll talk about that in the next slide. We have the nitrogenous base in this pentose sugar. This is DNA then. And a 5' end hanging off this 5' carbon, and this 3' end off the 3' carbon, and a sugar phosphate backbone, right? So a nucleotide means a nucleoside plus a phosphate group. A nucleoside is a nitrogenous base in a sugar. There are two families, the pyrimidines and purines, something I've already mentioned, and they have a 6-membered ring and a 6-membered ring fused to a 5-member ring. And again, they are connected by a specific bond in this group, and it's called a phosphodiester bond, and you do need to know that. So we have these nitrogenous bases right here. They look great. They're a lot of fun. You don't need to draw these. You do need to know the names and where they're found. So let's look at this deoxyribonucleic acids, and we're going to actually focus on some of the characteristics, and then we'll focus on this later in the rest of the class. So deoxyribonucleic acid, DNA, is going to encode for information for amino acids. So it's going to give us the code to the protein. The protein's the secret sauce, right? But we need to know what that code is. And so many years have been spent on studying what is the research, what is the genetic code, what's that secret sauce. So we do this by looking at DNA. And DNA is found as a double helix. You can see it in this picture here, and it has 2 polynucleotides, so many nucleotides lined up together to connect by hydrogen bonds. And we can see that we have complementary base pairing rules. A will be bound with T and C will be bound to G. And in the case of urine, it's not a double helix, and the complementary base pairing rules is A with U and C to G. Furthermore, in DNA, we have 2 backbones and we run a 5 to 3. It's really important you know 5 with an arrow to 3. This will help us understand directions, and we know that that 5', there's a phosphate hanging off there, and a 3', there's an OH hanging off there. And we know that when we draw that 5 to 3 and 5 to 3, that these strands are actually going to run anti-parallel. And so we will see this, and we will see that a DNA molecule is including many genes. Furthermore, we can see how the sugar phosphate backbone, it can be very complex. I don't like this picture a whole lot for a couple of reasons. A, double bonds to T, here it has 3 dots, so that's actually not really a good representation. C, 3 dots, C, triple bonds to G. So we will kind of talk about that more coming into the thing, but just know that this is great for the 5 and 3s, but it's not great for the dots. All right, and then we have ribonucleic acids, and it's going to be similar. It's going to contain ribose instead of deoxyribose and contain uracil instead of thymine. So here's a picture of DNA at the top, and then we have RNA at the bottom. And with these RNA, we're going to see that they have bases along this ribose phosphate backbone. And it's a single polynucleotide chain. And again, RNA is going to use this information to sequence amino acids into proteins. So it's going to be really helpful for that. So check your understanding. So a shortage of phosphorus in the soil would make it especially difficult for a plant to manufacture what? DNA. Which is the structural level of a protein would be least affected by a disruption in hydrogen bonding? Primary structure. And it's important that you know this. This is a good test question because when we look at what type of bonding occurs at the different structures, you do need to know that. So if there is a disruption in hydrogen bonding, it's going to be really backfire at the tertiary and secondary level. So then we have multiple charts and multiple things that students have used to understand how to fill out their chart and how to study. So I'm going to click through these. These are you can see the difference of the different types of 5' versus a 3' end. How can you create a complementary strand? What does that look like? Then we have a couple other charts. This is looking at the anti-parallel strand, 5 to 3, 5 to 3, right here, original strand, and then when we make a complement, it's going to be really important that you know that. That concludes this presentation.